Good morning, everyone. I'm Chiara Rossi, and I'm a PhD student from University of Georgia. And today I will present my poster, which is about the plant growth and leaf spot control of peanut plants treated with different fungicide programs. Peanuts are susceptible to many soil-borne and foliar disease that can affect negatively the plant growth and also the final yield. To reduce the severity of this disease, many fungicide programs with a wide range of control have been developed and used. However, there is still a lack of information about the effects of fungicides on the physiology of peanut plants. And that's why the main objective of this project was to evaluate the effect of different fungicide programs on leaf spot intensity and plant growth in peanut crop. This project was developed in Attapulgus, which is part of University of Georgia Research and Education Center. The experimental design used was a randomized complete block design with 5 replications, and the treatments were 3 different cultivars, Georgia 6G, Georgia 18RU, and TifNV-High O, and 4 different fungicide programs, non-treated control, chlorothalonil applied 5 times over the season, chlorothalonil applied 7 times over the season, and chlorothalonil applied 3 times plus Miravis applied 2 times over the season. The measurements that we took were leaf area index by using a septometer, leaf spot intensity using the Florida 1 to 10 scale, and pod yield at harvest. For the data analysis, we use ANOVA and Tukey test a 5% probability. I will now go over some of our results. On the top of the poster, the first graph on the left represents the effect of fungicides on leaf spot. The plots with the highest leaf spot intensity were the non-treated plots, while the one with the lowest leaf spot intensity were the one treated with chlorothalonil applied 7 times over the season. Moving to the next graph, figure 2 represents the effect of cultivars on leaf spot. The results indicated greater severity of leaf spot in plots planted with Georgia 18RU, followed by Georgia 6G, and lastly TifNV-High O with the lowest intensity. Consequently, leaf area index was greater in plots treated with these fungicides due to less defoliation, as we can observe in figure 3. This graph, in fact, represents the effect of fungicides on leaf area index. As we can see, the plots with a higher leaf area index were the ones treated with chlorothalonil applied 7 times over the season. Figure 4 represents the correlation between leaf spot and leaf area index. The correlation was negative with a R-square of 0.66 between leaf spot and leaf area index, indicating that greater leaf spot intensity resulted in lower leaf area index. Figure 5 represents the effect of fungicides on pod yield. The plots that yield the most were the ones treated with chlorothalonil applied 7 times over the season, while the plots with the lowest yield were the non-treated plots. The last graph, which is figure 6, represents the effects of cultivar on yield. The plots that yield the most were the ones planted with TifNV-High O, while the plots with the lowest yield were the ones planted with Georgia 6G. In conclusion, chlorothalonil applied 7 times over the season and the cultivar TifNV-High O demonstrated to improve the control of leaf spot intensity without negatively interfering on canopy leaf area index and pod yield. There are, however, future research that can be done, for example, to validate the results obtained in this research by conducting the same experiment in 2021. We actually already started to collect samples and data this season. Also, to conduct this trial in different sites to expose plants to different levels of disease severity. And lastly, to select other cultivars to evaluate the efficacy of these fungicides on leaf spot control and plant growth. With that, I would like to thank all my colleagues that helped me with this experiment, National Peanut Board for funding this project, and Georgia Seed Development for donating the seeds. Thank you.